where we applied, or rather we employed a machine learning approach. So ML stands for machine learning approach, machine learning. So where we had employed one of the machine learning approaches for undertaking these studies. So most of you may already know. If not, you will be knowing what are machine learning approaches. But nonetheless, I'll just try to give a very brief introduction to what we refer to as it is not no, what we refer to as machine learning. So machine learning is a subdomain of artificial intelligence. So artificial intelligence, we all know. Even if you ask uh, you know, a commoner, he would be able to say what is artificial intelligence. So essentially, it is a computer-assisted mimicking of human intelligence. So if you carefully look at our human intelligence, or rather human intellect, it learns through examples. So we are not inborn with ready so-called ideas about human race, various subjects, maths, physics, chemistry, just as an example, or the countries, their various populations, etc., etc., various types of animals, etc. So we all learn during our lifetime. So that is through example. So if you consider, for example, if we see an animal and label that as a lion, we have actually seen many lions, during our growth period, and we have collected enough information in our brain to label a given animal, given its features, that it is an animal, it's a lion. So similarly, if we are shown another animal, perhaps we may label that as a tiger or donkey or horse or some human being. Even among human beings, we may label them as Indians, Africans, Mongolians, Caucasian, something, etc., etc. So this kind of human intellect, it is there inherent in all of us to various extent. So this capability arises essentially because of we learn through examples. So we don't really consciously do it, but what people say, it's not even unconscious, but somehow we can say semi-consciously we learn all these things through our life. So this is what the artificial intelligence tries to do. It tries to mimic the human intellect of learning things, learning through examples and labeling them, or rather identifying to a given category, right? So how do we do artificial intelligence, or if we more specifically speaking, machine learning? In fact, machine learning is a subdomain of artificial intelligence, according to some authors. Some people say that it is an extension of statistical learning. In statistics, if some of you know, so we through regression we try to build models. For example, if you have a scatter of points in two-dimensional space, x, y, x and y plane, a scatter of points. So we try to fit a function describing the relationship between the points. For example, we may fit a straight line with certain slope and certain intercept. So that actually becomes so-called, a least square fit. Least square fitting is essentially a mathematical modeling of, how the data points are connected to each other in a linear fashion. So therefore, we try to get the slope and intercept, and that actually becomes our model as far as that, scatter of data points is concerned. So similarly, so people say that the machine learning is an extension of statistical learning, and some people say that it is a subdomain of artificial intelligence. So essentially, machine learning, what do they have? They comprise of some computer algorithms which are applied to set of features set of features. So this is very important. Where are they arising out of? They are arising out of some samples and examples that belong to a certain category or a system. For example, if you are talking about an animal called a lion, now, so you have certain features associated with lions. For example, their skin tone, the kind of the head size, the way they roar, the way they feed, etc., etc. So you can consider various things as features representing that set of lions, right? So computer algorithms are there for learning about the systems in question from the features that characterize those that system. So in computer, when we use computer algorithms, we say we are trying to build a model or trying to train a model corresponding to a system under question or under investigation. So essentially, what are we machine learning approach? It's essentially a computer algorithm, a computer approach, computational approach, which essentially builds a model or trains a model for a given category of a system by means of the features extracted from, or that the features that characterize a system. Now, so if we consider the present, topics that I am going to explain to you, so we, as I said, we are going to deal with protein folds. That is how to identify protein three-dimensional structures from their sequences, right? So if we want to know something about protein fold, or to characterize a protein fold, we have in our hand their amino acid sequences, that is primary structure, what we refer to as. We have secondary structures, etc., etc. So various kinds of sequence and structural features we could compute from the sequence. If we know the structure, we could compute and then try to identify what kind of a fold that protein has. The second example which we can think of is what are missense mutations, right? So I was told that most of you are biotechnologists, so therefore you know what is a missense mutation. If not, I'll define what is a missense mutation during this talk. So there are mutations that are actually seen in DNA sequences, and the mutations happen randomly, and there are repair systems to do to remove such any aberrant changes in the DNA sequence. But if for some reasons some changes escape those repair systems, and they are they get recorded in the DNA sequence. So DNA sequence, when it is translated to protein sequence, might result in a change in the amino acid residues. So if there is any change in the amino acid residues, that is what is we refer to as a missense mutation. In fact, these are point mutations in DNA which lead to changes in amino acid residues. So some of these mutations are well accommodated within the protein structure and function. So they may not lead to an onset of disease phenotype. Mutations do. Those have been discovered in populations by through genetic screening, and the people have identified mutations and which seem to be associated with certain diseases, and therefore they are referred to as disease-causing mutations or disease-associated missense mutations. And many mutations, even though they are missense, but they cannot be associated to any disease. Such mutations have been referred to as benign. Given a missense mutation, its functional impact as whether it's a disease causing or it's going to be benign, right? So from using the missense mutations sequence and structural context, So these two examples, they come under the purview of, or rather the challenges that could be dealt with using the so-called machine learning approach. So that's what I'm going to talk, in this lecture. Now, the one of the important requirement of conducting any machine learning, uh, important requirement is having sufficiently large number of samples or examples to train a model. So, for example, the fact that you have developed a, a model for a lion, an animal, to be labeled as a lion, you must have actually seen sufficient, lion vis-a-vis the other animals. So therefore, actually, you can discriminate between, for example, a lion and a tiger, or a lion and a cow, lion and a human being, etc., etc. So you are not learning the features that characterize an animal, but also you are trying to learn the discriminatory features. So that distinguish between lion and human being, lion and horse, etc., etc. So, but in order to have a intuitive model, right? So you need to have sufficiently large number of samples and or examples of a given system or a given category of a system. So in fact, this is one of the bottlenecks why machine learning approach cannot be used for all the examples that we see in biology. So in biology, if you see, being a biotechnologist, you know that number of samples, good enough number of samples is always a bottleneck. For example, if you want to learn, if you want to conduct some cancer research, so you need to collect some samples from patients, say some tissue samples, and the number of samples you may collect may run into few numbers, maybe 10, 15, 20, like that. But that may not be sufficient if you see really, when you consider the population-wide study. Anyway, so what are the types of machine learning approaches we have, right? So the first one is what is referred to as unsupervised learning methods. Example is principal component analysis. In short, we refer to as PCA, principal component analysis. So unsupervised learning. So that is one category of methods we have. And the second category, the other category we have among MLs is supervised learning methods. So example is support vector machine, SVM. So these are some of the some titles may look little uncomprehendable, but just ignore for time being. So principal component analysis, if you are not really aware of this, this may look very alpha, beta, gamma type of sound, and support vector machine. So what is vector machine? What is support, etc. So they may look little hi-fi, but just ignore that immediate reaction, but just here. So what do we mean by unsupervised learning and supervised learning? So as I said, machine learning approaches are two examples, and examples, essentially speaking, some characteristic features describing those examples for a given category, right? A number of lions, that is examples, say 100 lions is number of examples, and the features that characterize the animal, such as lion, right? So there are various features you can think of. Okay, this is so when we look at, for example, if you collect the features of lions and features of tigers, right? So you don't really call those features if you do analysis, for example, using this so-called principal component analysis. They may naturally cluster, as two distinct clusters. The set of features that are describing the lions and the set of features that are describing tigers, they may inherently emerge out as two distinct clusters. So there we are not really calling one cluster as that of a lion, the other that of tiger. So this is what is referred to as unsupervised learning. The supervised learning approaches are where we say these is arising out of lion. These features are arising out of tiger, right? So like that, we are giving labels to the set of features. So whatever the discrimination or the division between or the differences between the two set of features, so that is what is referred to as supervised learning approach. So these two methods are available. So in my case, whatever I'm going to speak on, so we will be using supervised learning method, or rather we have used supervised learning method. More specifically speaking, support vector machines. Now, there are few jargons in machine learning, so I'll try to introduce. So, as I said, a machine learning approach essentially learns or trains a model through examples, or more specifically, features extracted from examples. So, therefore, we define what are referred to as sets, data sets. So, there is a set trainings. So these are the set of examples or samples and their features, okay, that characterize that whatever the category we are thinking of. So these samples or examples and the features extracted from those samples and using which we train a model that becomes a training set. For example, you are talking about uh, building a model or training a model for globin superfamily of proteins. So you train the model using the protein sequence features and structural features, right? That characterize the globin superfamily. Next is the test set. So having built a model or having trained a model, so these are the jargons that are used right in machine learning. So you have to validate the model. So how accurately that model can reproduce results. Suppose if you if you don't know what is the fold of a protein, but you have some sequence and structural information from those set of proteins. So use those set of protein sequence and structural features and give it to the model. And as model, what is the protein fold? It's a prediction. So having trained a model, you are asking the model to make a prediction. So in the training, what are we doing? We know what are the samples, and we're using the features that are extracted from those samples to build a model, build a picture. Now you are using the picture or the model and trying to get some features from an unknown entity, and then try to see whether that unknown entity indeed belongs to the system or for which the model was trained, right? So either the model can say yes or no. Yes, the features are such that the features are associated to, for example, lion or tiger or whatever model that was built. So so in that we use what is referred to as a test set, where we know what is the label, but we are trying to pretend we don't know the label, but we are trying to predict the label, right? So through some another set. So this is what is referred to as a test set. Now, this is how a model is validated for its prediction accuracy. There is another jargon that is used is what is referred to as a model validation, where typically we, if suppose you have uh, trained a model using say 100 samples, now you have tested the model using another independent samples, then you found the accuracy. But that is not sufficient, because you might have trained using a set which might have certain biases, and when you are testing it, it may also have its own biases. Now you want to remove the biasness in from the training as well as testing. So essentially what you do, essentially you club both the sets, um, and then divide the two sets into two, where you consider one as a training set, the other as a testing set. Now you switch, right? So the test set becomes the training set in the second cycle, and the training set becomes a test set. So this is what is, and then measure the accuracy of the prediction. So this way you are, and then take the average of two predictions, and that you represent as a prediction accuracy of the trained model. So this is what is referred to as a cross validation. So what we did, we actually divided a given set into two parts. So therefore, it is referred to as twofold cross validation. Suppose you have enough number of examples, you can actually divide the entire data into 10 equal parts. One, you use it as a test set, and the remaining 9 as training sets, and then get build the model, then test its accuracy, etc. You can, and then in the next cycle choose the another set, another part, 10 sets as the test set, and the remaining 9 sets as training set. So you can actually do this 10 times, right? So this is what is referred as 10-fold cross validation. So depending on the the size of the data set, you can be more ambitious, like 20-fold cross validation, etc., etc. So really people go up to 10-fold cross validation. So that is the that is what is in vogue or in practice. Now, so what machine learning method that we had used in our studies, right? The two studies I was mentioning. So the machine learning approach that we had used was is referred to as support vector machine. It's a binary classifier. So it was actually developed by a mathematician called Vapnik, or computational scientist, you can call him, in 1960s. But it started extensively, its use was extensively done in 90s. So it is actually a classification method. It's, in fact, specifically speaking, it's a binary classifier. So suppose you have a collection of points like this, right? So you have collection of red points and collection of blue points in a in space, for example, as shown here in two-dimensional, as a two-dimensional plot. So it so happened that all the red dots may segregate in one region, all the blue dots may segregate in another region. So the support vector machine, it actually tries to define a plane or a boundary, right? So that divides or discriminates the two sets of points, right? So using what are referred to as support vectors. So the points, so it doesn't use all the points as it is done in regression analysis. Rather, it chooses only a subset of points, what are referred to as support vectors. Okay. So the machine means it's a machine, it's an algorithm. So it tries to build what is referred to as a hyperplane that stands as a boundary between two sets of points, two distinct set of points. So here graphically they are represented as blue points and red points. So as you can see, this line clearly or linearly separates the two sets of points, namely blue and red. So this is very ideal situation, but the features that we use may so happen that some of the red points, I mean, just as an example saying it, they are not red and blue always. So it could be other features, right? So some of the red points may be here and some of the blue points may be here, right? So therefore, you may have to give some allowance as far as the boundary of this hyperplane is concerned. So this is as hyperplane because this happens not in 2 dimensions, it happens in multi-dimensions or hyperdimensional space, what people call it as. So some allowance may have to be given. So what are the size of the boundary, right, as shown here, right, dotted lines here about this solid line. So you can actually choose the size of so-called the interval, or you can say the size of the boundary, such that some allowances can be given. Some blue dots may fall within this, and some red dots as well. The separation, the size of the separation or the linear separation could be a trade-off between how many misclassifications you may do, right? But if you consider the number of samples that are getting classified, right? So they may be sufficiently large. So some allowances you can give as far as misclassification concerned. So therefore you can set the boundary. So these I mean, I'm oversimplifying rather. But this is how an hyperplane is constructed using a support vector machine. So for this, we need not actually worry about whether should I write a program, what maths is there, etc. So that we need not worry. So there are packages available. For example, if you are aware of R programming language, so there are functions for doing these calculations, support vector machine computations and building models. Like there is a suite of packages referred to as libsvm. So that also gives access to usage of support vector machine algorithm. So all you need to do is you have to supply a set, as I said, characteristic features, given a label, and then ask the system to develop a model. So now we'll come to the biological question. So far we were only worried about what is a machine learning approach, what methods are available, and specifically speaking, one machine learning approach referred to as support vector machine. So now coming to the question of application of machine learning approach for protein 3D structure prediction. Now we all know how important protein structures are. That we came to know from an experiment. In fact, that was a very famous problem referred to as protein folding problem. So Ian Pinson in 1961, he conducted an experiment, in fact, on ribonuclease A. He unfolded the protein. So he actually set denaturation conditions such that it is actually this ribonuclease A is stabilized by 3 disulfide bonds. And when denaturation conditions were set, these were disrupted, and therefore the protein gets unfolded, unfolded. So, and when he set the conditions for favorable for refolding, it readily refolded. So it was an experiment of protein unfolding and refolding. So this experiment, what he conducted, it was published in PNAS 1961, that fetched Nobel Prize in 1972 for Ian Pinson. So from this, it became very clear for scientists that the force for a protein to fold into its native three-dimensional structure is if not entirely, but mostly hidden in its primary sequence or primary structure or the amino acid sequence. So that what they referred to the so-called the problem of protein folding, right? So that also led to what is referred to as now famously protein sequence structure function paradigm. Okay, so that means a protein is characterized by its amino acid sequence. Because of its sequence, it adopts a native three-dimensional structure, and its function, molecular function, what we refer to as a molecular function, is dependent on its three-dimensional structure. Suppose if you disrupt its sequence to an extent that its three-dimensional structure is disrupted, then its function is mostly gets affected. So this is very famously referred to as protein sequence structure function paradigm. So which essentially tells us that 3D structures of proteins are very essential for better understanding of their molecular functions. Now, so how this information of three-dimensional structure, so there are innumerable examples in literature where people have actually used protein three-dimensional structural information to design drugs. So one case study is, with respect to development of an inhibitor for HIV protease. So this is the three-dimensional structure, a representative three-dimensional structure of this protease. It's a crystal structure, and using this crystal structure, they found out the binding site of this protease, and then they designed an inhibitor that can best fit the best sits within the binding pocket. So using utilizing the interactions that are available within that binding pocket, so that it can tightly bind there, and cause the competitive binding effect, and therefore stop the natural ligand or substrate. Right. So this is one of the, like that you have innumerable examples in literature. Right. So all it tries, I'm trying to say, is the importance of knowledge of protein three-dimensional structure. Now so we now spoke so far about protein sequence structure function paradigm, but the story doesn't end there. We have a set of proteins which are referred to as in literature intrinsically disordered proteins, or IDPs. So these IDPs are enriched with so-called intrinsically disordered regions. So what are these intrinsically disordered regions? So if you try to see the three-dimensional structure, you cannot see them by means of experiments like X-ray crystallography, NMR, spectroscopy, etc. So rather, you will see as a blob. So why? Because these regions, they quickly adopt structures, and they in fact move from one conformational state to the another conformational state at a very quick time scale, such that the experiments can't really record them as one snapshot. So, for example, if you look at a moving fan, you see as a disc. You can't really say how many blades are there, what are their shapes, sizes. In fact, how many blades are there? You can't really make out if the fan is fast rotating. So similarly, you can't really make out what kind of one three-dimensional structure or two-dimensional, whether a protein or a region in a protein is forming one unique structure or many unique structures. You can't really see experimentally. So such regions are referred to as disordered regions. In fact, it has been estimated that 1/3 of human proteome is actually made of proteins which are enriched with these intrinsically disordered regions. In fact, there is a whole lot of literature on these so-called intrinsically disordered proteins and how important they are biologically, etc., etc. In fact, if you look at the abundance IDPs, they are there in archaea, bacteria, eukaryotes, even in humans. You have so many. Especially even if you look at the alternate splicing mechanism in human that impact the human protein translations. So, in fact, we have found even the major isoform may be a stable protein, but some of the minor isoforms, they tend to become intrinsically disordered proteins. So there's a whole lot of story as far as protein structure is concerned. Now, so as a consequence, there is now an emerging paradigm, which is referred to as protein sequence disorder function paradigm. In fact, many interesting stories have been increasingly reported in the literature now. So that is about the importance of protein. Okay.
Sorry to interrupt you, sir. Shikha Singh, ma'am, can you please put your mic off?
No,
Shikha
that's
Singh,
okay.
ma'am.
So there is an young participant also there. That's fine. So that is one story. So far we know how important the 3D structural information is about. Now, if you look at the number of protein sequences or the number of proteins for which only we know the sequence information, vis-a-vis the number of proteins for which we know their three-dimensional structure. So there is a huge difference in the number. So this graph is actually, plotted some time ago in 2008. In fact, the story has not changed much. Even now, if you just plot the number of, the line that is shown almost parallel to x-axis, that is the graph depicting the number of protein structures available year-wise. And this line represents the number of proteins whose only sequence information is available, right? So if you can see this, there's a huge gap. So this means to say that we have large number of proteins for which we only know sequence information, but we don't know their three-dimensional structure. So this is what is famously referred to as protein sequence structure gap. Okay. In fact, there are so many enzymes for which we don't know the three-dimensional structure. So knowing the three-dimensional structure is very very important, because if you want to think of designing drug as a rational approach, the starting point is actually the three-dimensional structure. Anyway, so how do we now predict protein three-dimensional structure, at least at the level of what could be its fold, right? So what is referred to as, that challenge is referred to as protein fold recognition. So what is protein fold recognition? It's protein fold recognition is a computer program residing in a computer, as shown here. So input is your protein sequence, whatever the protein sequence for which you wish to know what kind of protein fold it may adopt, right? The output would be a matching fold. So how this black box works, the computer program works, it has a library in its in its database, a library of known protein folds. So the given sequence is aligned to each of these protein folds, okay, in the library, and their compatibility or the alignment compatibility is calculated for each of the folds. So whatever the fold that gives the highest compatibility, that is actually given as an output or as a predicted fold for that input sequence. So this is essentially what is referred to as protein fold recognition. Now you may ask a question, why protein fold recognition, right? So you all know that protein structure, perhaps you all know that protein structure is most conserved than sequence during the evolution, okay? So this is a very well-known fact. And if you look at the number of protein structures that is available in a database referred to as protein data bank, So there are about 1,000 different protein folds. All the known protein structures, adopt one of the 1,000 protein folds. So that means we know 1,000 different protein folds, right? And people have been trying to estimate how many folds are possible in nature. So at different points of time, people have come up with different numbers. So if we consider this reference, which published in 1999, so Govindarajan et al. So they estimated in nature, right? If you consider all the protein sequences belonging to all sorts of, plants, animals, bacteria, fungi, etc., etc. So they adopt one of the 2,000 protein folds. So we already know 1,000 and 2,000. So therefore, if you take randomly any protein sequence, there is a 50% chance that it adopts one of the protein folds, right? So that is the probability estimate. So given this, therefore, a protein fold recognition method becomes very attractive because there is a 50% chance that your protein that you have tested, adopts one of the known protein folds. Now, in order to make the protein fold recognition or fold prediction, the machine learning methods have been, chosen as attractive methods. So in order to, as I already said, machine learning approaches require sufficient number of examples to learn. So do we have sufficient number of examples, right? So that is a question. The second question is, do we have a gold standard where you can train a model, test the model, and publish your paper? You have to publish a paper after all. So there is, in literature, there is a gold data set that was given by Ding and Dubček. So these are the two authors, and therefore it goes by their name, data set too. It was actually published in 2001. So this data set contains these many, proteins, 311 proteins and 383 proteins, which are one set is used for training and the other set is used for testing, and this corresponds to 27 folds only. Okay, so but this is used as a gold standard for testing your method. So how this training and test set was compiled by these two authors. So in the training set, they made sure that no two proteins have more than 35% identity, and so therefore all the proteins that are there in each of the 20 folds, they are so-called sequentially distantly related, so to say, but they adopt similar protein fold, right? So there is no sequence biasness, right, as far as the fold is concerned. For example, if you consider a fold which is having 100 sequences, all of them are 95% or 90% similar, then there is a sort of a bias you are introducing into the training set. Then if you use a protein which is remotely related but might adopt the same fold, but that would not be predicted as that fold because it is overly trained for highly similar sequences. So in machine learning approach, it is referred to as overfitting model, right? So what features could be selected? So we selected. So important is amino acid composition. So you have 20 amino acids. So what is the frequency of each of these amino acids, right? So there is something dimensionality that let me not confuse you with this term. And the second set of features, what is referred to as dipeptide composition. So it is amino acid composition is the single letter composition. There are 20 amino acids. So for each fold, given the number of sequences or number of samples in each fold, you can compute what is the percentage of alanine, cysteine, proline, etc., etc. Similarly, you can form the amino acid pair, so-called dipeptides, and their composition, meaning what is the frequency of seeing alanine, alanine, ala together, or ala pro, or pro, etc., etc., right? And there's something we introduce what is referred as second order amino acid pair. So you can think of a dipeptide, but with a gap. So for example, A followed by any amino acid followed by alanine. So that is AA dipeptide, but with a gap, right? So these are the three sequence features we used. And then we also used some structural features. So you know the secondary structural states, namely alpha helices. Alpha helix is one of the major class among helices. So helices, beta strands, and loops. So you can then calculate the frequency of amino acids occurring in these three states. That is one. And dipeptide frequencies occurring in these three states, right? And the secondary, dipeptide frequency, that is the dipeptides with one gap, what is their frequency? And the second category of structural feature is the burial status of amino acids in a protein fold. So whether a given amino acid is buried or exposed, right? So all these things are computed from so-called DD data set of certain three-dimensional structures are known, of course, sequences are known, and you calculate all these features. So for calculation or identification of secondary structural information, we could have actually used the known structures, but rather we tried to use the predicted values for the secondary structural information, like helices, strand, and loops. And similarly, we use the predicted solvent accessibility information, right? So why we used predicted values? Because we wanted to use the ones which are known to be associated with a fold, but their three-dimensional structures are not known. So thereby we wanted to increase the number of samples which we could bring into this fold of prediction. Now, as I said, we have used support vector machine, which is a binary classifier. But if you look at the protein folding problem, it's a multi-class prediction. In the sense, if you are given a protein sequence, there are a number of protein folds which need to be tested against, right? So which say like 28 or 1,000 or 100, whatever the number of folds you are having in a library, right? So you have to use each of these your amino acid sequence against each of these folds, and see which fold is the most compatible one. Here in the machine learning, we are actually building model for each of the folds from their examples, right? So each fold gives rise to a model, right? So it's a multi-class prediction, but SVM is actually a binary classifier, right? So how do we do that? There are approaches. So let me not go into the details of this. One versus all, that is a method, and one versus one. So briefly speaking, so you can think of creating a support vector machine model. For example, there is a globin superfamily, and all the remaining superfamilies or folds you can consider as the second set. So one set is globin fold, the other set is all the other folds. Okay, so you train the model for that. Okay, so that means you are training a model with one for globin and for the remaining folds. So that is what is referred as one versus all. So that you have to do for all the protein folds, right? So protease family one against all the remaining folds. So like that. So one versus all. right? 1 versus 1 means you have to take a pair of protein folds and train so many machine learning models, right? So if you have 10 folds, so that means 10 into 9 by 2. So those so many pairs of models you would be generating. So where a given sequence is fed to each of these models, whichever comes out as it's called selection by voting. So whichever model gives the maximum predictions, so that is selected as the predict as a fold. So not only that, so this
Sorry to disturb you, sir.
yeah.
Sir, if you wish, we can have a break of 5 to 10 minutes.
I'll just
Yeah.
complete. I'll just take 1 minute, 1 or 2 minutes. So I think almost end of first part.
Okay,
Okay.
okay, sir. Okay,
Yeah.
sir.
So we, in fact, that is not the end of the story. We actually built what is referred as hierarchical approach for protein fold recognition. Because as I said, because of the number of samples that we have, and also the complication of binary extending binary classifier into multi-fold classifier, so there was an inherent drawback or the bottleneck. So what we did a trick. So just like we developed models for protein folds, we also developed models for protein classes. So, if you look at the protein structures, so they all fall into 4 major classes. One major class is where all the protein folds are predominantly having helices. That is referred to as alpha class. Then there is another class, alpha beta class. So in that itself, you have alpha / beta class, alpha plus beta class. So these are the two. That means the proportion of helices and beta strands, they're almost like 50%. So alpha / beta means there is alpha followed by beta followed by alpha. So like that, if you go from sequence from N-terminal to C-terminal, you see alternating helical and beta strand structures, right? And alpha plus beta means so that protein fold has one set of helices followed by one set of beta strands as you move from N-terminal to C-terminal. So they're actually two subdomains. And the fourth category is beta class, where the entire protein sequence is mostly comprises of beta strands, which aggregate to form beta sheets. So alpha class, alpha plus beta class, alpha / beta class, and beta class. So you can actually build classifiers for these 4 categories. So once you identify, given a protein sequence, that it belongs to one of the classes, then you can actually create another layer of models where you can get multi-classification multi-classifiers within the alpha class, within alpha plus beta class, alpha / beta class, and beta class. So that way we were able to bring many protein sequences into the fold of protein fold prediction using machine learning approach. So this is the last slide as far as this part of the talk is concerned. So what we found, so what is our take-home lesson from this? This is a way, what we found that the sensitivity and specificity are not the same for all the folds, right? This is the accuracy is related to the prediction accuracy, right? So in general, AlphaFold class was, predicted with better sensitivity and specificity than beta class folds. That is mainly because alpha helices are better predicted than, beta strands. So that is actually the, inherent drawback in secondary structure prediction algorithms. And therefore, the protein folds where we could not get good sensitivity. So they were mainly because of the errors in their, in the prediction of secondary structural states and also the solvent accessibility state. So some protein folds, either because the number of examples that we could get were, small, and therefore the learning was not complete. So therefore there were some inherent, errors. And so this entire thing is available, this entire server is available. Sorry. Oops. Fertilizer part, anyway. This should work. I don't know why it's not working. So this is the website address. So if you can access this web server, it's available on the public domain. You can test for yourself the protein fold. You can take any protein sequence and submit, and then you get a prediction for its three-dimensional fold. And these are the published papers for this work. So we'll take a break and then we'll come back and then look at the second topic. Thank you very much.
If you wish, I can share on your behalf the. So just a minute, sir. Let me.
This one, the server.
Yes, sir. The server.
Yeah. You can show. No, it should not work.
Yeah, here it is. Sure. So here it is.
Yeah. So it's very simple. All you have to do is you have to give the name for your sequence, email ID if preferable, or it can show online, right? If you have the sequence in a FASTA format, you have to choose the file. I mean, this I need not explain, or copy and paste into this window and submit sequence. So it takes some time to get you the prediction, right? And also there are many other tools that are there for fold recognition, right? So you can use any of them. So this is the server that is available. Yeah. So let us come back after some break.
Sure, sir.
So how much break we have?
Sir, as per your convenience, so we can have 5 to 10 minutes break.
Yeah, actually I can continue and complete.
No problem, sir. Or if any of the participants have
Shall I
any
continue or shall we
till
break?
this part of the session, one can either put their questions on the chat box or can ask questions directly to sir. If any of the participants have any kind of.
I'm actually more comfortable with Google Meet than this Zoom. Where is the chat box here?
Sir, it is
here, yeah, correct.
Yeah.
I don't see any chat there.
Yeah, participants, if you have any kind of questions or queries related to the first part of our session today, you can just simply ask your questions directly to sir, or you can just write in the chat box. So there is a question from one of our participants.
Yeah. How 1 versus 1 algorithm for protein fold recognition works? No, this is just an extension of binary classification to multi-classification. So what we did, test. In fact, it's published work. So we found that there was no, difference as far as the prediction accuracy is concerned between 1 versus 1 and 1 versus all algorithm as far as the prediction accuracy is concerned. So there was no, so you could use either of the one. So in fact, I forgot what was the, I think we have used, there is one more method called Kramer's method. So where it's a very slow process, but it tries to build a multi-model by optimizing for all the protein folds. I think we have used, but we didn't find any difference as far as prediction accuracy is concerned.
Okay, sir. Okay. Thank you, sir.
If any of the participants have any query, can ask. There is one more question, I guess.
Oh, okay. So how is FPFP server is different from other servers like Fire2? That's a very good question. Okay, so this machine learning approaches are referred to as taxonomic methods. That is a word that is used. So the purpose of this machine learning approach is only to identify okay, given protein sequence, this is the fold, right? So it doesn't give you any alignment. It doesn't give you in Fire2. If you see, if you submit any protein sequence, you not only get the alignment, right, of the whatever the known protein, known, say a protein fold is known, some example is there with the structure, so you get an alignment of your input sequence with the closest protein structure that is representing that fold. So you also get sequence alignment there. In fact, Fire2 also gives you a model, okay, three-dimensional model as well, constructed using that so-called the closest protein structure. So that is they are fold recognition methods, but they use what is referred to as sequence to structure 3D to 1D profile or profile scores. That's a different method. But this machine learning approach is a different one. So you are building a model, you are just trying to see which is the best compatible fold, or which is the model that is that could be best fit given its characteristic features. So ideally speaking, you just get, okay, this is the protein sequence, okay, whatever the name you are given, and this is the most compatible protein folds. That's it. So there is no alignment, there is no three-dimensional structure available. In the server that we have built, we actually provided a facility where a fold is predicted, and a representative protein is taken from that fold, and then an alignment is made. So we have provided an extension, but in in a machine learning approach, that is, so you don't get the alignment, right? You only get the prediction. That's it. So that is a distinction between Fire2 and then, for example, HPFP. Okay, how to relate HPFP with AlphaFold algorithm? AlphaFold algorithm is a what people say is a deep learning approach. So they have used almost all the known protein structures, and they have used artificial intelligence. What they say as even I have not read the paper completely, but whatever I know, I have read. So AlphaFold is actually, it gives you three-dimensional structure. It is like a homology modeling tool. You know what is homology modeling. So homology modeling is an approach where you use a known structure as a template, and use the alignment, sequence alignment of your protein for which you want to build a certain structure with the template structure, and try to build a model. So when we say try to build a model, we are trying to get the positional XYZ coordinates of each of the constituent atom in the protein that you wish to build a model. So it is like determining three-dimensional structure. So what do you mean by determining three-dimensional structure by X-ray or NMR spectroscopy or cryo-EM? So the idea is to get the positional coordinates of the constituent atoms in the protein, right? So what is alpha carbon position, what is the hydrogen position, what is nitrogen position, or at a higher level, what is the position of this residue, that residue in three-dimensional space. So that is what is referred to as 3D structure. Either you determine the three-dimensional structure or you can predict three-dimensional structure, right? So prediction, the best method available is homology modeling. So AlphaFold does that kind of homology modeling. HPFP doesn't do homology modeling. It's only prediction of a fold, right? What is the fold? That's it. There is no prediction. There is no three-dimensional structure that is actually computed. Okay, that's so that dusting the is PCA dimensional? Yes. Principal component analysis is the dimensionality reduction techniques, right? So that is yes. Okay. Thank you.
Thank you, sir.
In fact, AlphaFold is the, in fact, people have actually done, in fact, they have constructed database for all the, protein sequences there. In fact, ever since AlphaFold became available, all these fold recognition tools and everything have become now, taken backstage. So so that is the truth, right? So AlphaFold, now it does with great accuracy. In fact, you must be aware, or you should know, that there is a challenge that is done every alternate year or every 3 years or so. That is called CASP, Critical Assessment of Protein Structure Prediction. It's a challenge. It's an open challenge. So what this challenge is about? Suppose there's a committee for this purpose. So all the motive is to identify the best protein structure prediction tools, either homology modeling approach, fold recognition, or secondary structure prediction, etc., at various levels. So the idea is to identify the best prediction tool. Okay, so how it is done? So they contact the experimentalists like X-ray crystallographers, nuclear magnetic spectroscopists, cryo-EM people, etc., who are about to publish their protein three-dimensional structures. So the structures are not yet made public. Okay, so they collect all such protein sequences and pose it as a challenge. So people who wish to participate in that challenge, it's an challenge. So they have to apply their tools and then predict the three-dimensional structure, secondary structure prediction. In fact, even docking predictions, everything. So there are various types of prediction challenges available. So they have to do this and submit to that committee, and that committee evaluates the accuracy of the predictions in the light of experiment, determined structures. So that critical. So this, I think it used to be done once in 4 years. I'm not following that, any further. So using which people actually have identified which are the best tools available on the, at that particular point of time. Now, in the present, present stage, AlphaFold second version, I think it is the best prediction tool that is available, right? So that has surpassed all the tools. So if you think of HPFP, Fire tool, Modeler, whatever you think of, so it has surpassed all prediction approaches or methods. So now AlphaFold 2 is the best method available for the prediction. I suggest, if you ever want to predict the three-dimensional structure of protein, so just do AlphaFold. That's it. So we'll continue with second set. So that will be, I promise you, that will be very short.
Sure, sir.
Shall
No
I go
problem, sir.
ahead? Okay, yeah. Now we'll come to the second part, which is prediction of the functional impact of sense mutations in a human protein. For some of you, I just would like to explain what is a sense mutation. So as I told you, the mutations are random events, so they just happen. Sometimes this replication process may introduce some, you know, insertion, deletions, or substitutions. These are referred to edit operations, and that is what happens during the evolution of, sequences. And most of these mutations are repaired because there are repair mechanisms, there are repair enzymes, but some mutations might escape. Of these so-called point mutation, that is single base mutations, so the ones which lead to a change in the code, for example, CAT is codes for histidine, and instead of A in the second base position, A to C, and that leads to a codon CC2, which codes for proline. So when this piece of DNA gets translated into protein sequence, in place of histidine, you have a proline. So this is what is referred as a missense mutation, right? So as far as this so-called original protein sequence is concerned, this is actually an incorrect amino acid, right? So as far as the amino acid sequence is concerned, but this incorrect amino acid, is it really is accommodated within the structure or function of the protein? That is a question, to be answered. So anyway, so kind of changes in amino acids, right? So some are well accommodated within protein structure and function, and therefore the impact is not that much. So there is no perceivable impact at the molecular level itself, and therefore at the phenotype level. So you must understand that there is a relationship between genotype and phenotype. You are all your biotechnologists, you know very well. At the molecular level, whatever changes may happen, either they may get, translates into certain impairments in the downstream effects, and therefore a different phenotype may happen, which we refer to as a disease phenotype or whatever. So the missense mutations, right? So some of are well accommodated, and therefore they become neutral, right? As far as they're concerned, either at the molecular level or at the whole organism level. And some are not accommodated, and therefore they are referred to as deleterious mutations, which may, sometimes this human system, living system, has a lot of redundancies, right? So if one pathway, another pathway may be there. So therefore, even at the molecule, if the mutations are not so impactful, but eventually at the phenotypic level, there may not be any perceivable impact. I mean, these are all the various arguments, things happen. I mean, these are some of the observations which have been discovered time to time. So, but nonetheless, so you can think of two types of missense mutations: neutral and so-called pathogenic or deleterious. Now the question is, how do we identify, right? So people have been discovering through genetic screening whole lot of so-called missense mutations. For example, one example I am giving you here, there is a human CYP1B1. This is a cytochrome P451B1, right? So this is actually involved in the development of trabecular meshwork in eye, So if there are people have actually identified certain mutations in this protein that lead to so-called primary congenital glaucoma, PCG. So the primary congenital glaucoma is actually an inborn disease, and if it is untreated surgically, children may lose eyesight even before they become 3-year-old or 4-year-old, etc. Right? This is an inborn disease, and so many mutations, missense mutations, have been discovered. And so these are all disease associated or disease causing mutations, right? So, but these are known because these have been discovered from patients suffering from PCG, right? But the people have actually been discovering from 1000 genome projects or or simple genetic screening of population, healthy population, there have been discovered large number of missense mutations, right? So, but we don't know what are their functional impact. Are they disease causing? Right? So anyway, definition of healthy individual is a contesting, matter. So who is healthy? It's subjective. Anyway, so if we look into the literature, there are many tools available, and each one gives prediction at various levels of accuracy. So initially when we started this work, when my PhD student joined me, so the question was, we didn't know that time that there are so many tools available. So we just asked, can't we develop a method to predict pathogenic effect of unknown missense mutations? But after doing some literature search, we came to know that already some methods are available. So we slightly changed the question. Can't we develop a method which does better prediction than available methods? So we changed the slightly the question. So if you look at this question, so prediction of deleterious or not of missense mutations, it's a typical binary classifier, yes or no, right? Only two outcomes. So therefore, this problem fits very well, as compared to fold recognition, very well into this binary classification problem, and therefore we could use support vector machine, right? And as I said, we have some features which are characteristic of disease and neutral mutation. So what features we can think of? So one feature that we can clearly think of is the conservation or not so conservation of an amino acid at a given position in a protein family. For example, this is the sequence alignment of a certain protein family. These are the sequences, members in that protein family. And if you look at this position, you can say that G is completely conserved in this position. Then, so by virtue of this alignment, you can then argue that G is evolutionally conserved. Suppose if you see a missense mutation where G is, at the G position, you have a lysine K, then you can, it implies clearly through evolution argument that position that is highly conserved for glycine is now substituted by lysine, and therefore this if this position is structurally or functionally important, therefore you can say that the substitution is harmful as far as that protein as a molecule, right? So it could impact itself. Excuse me?
Sir, your slides
are not visible.
Oh.
After the last session, you just forgot to share your screen again.
Oh, is it? Okay,
yes,
sorry.
Sir, yes, sir.
But how did I come out of share? Okay, how did I come out of share? I did not unshare.
Actually, sir, I started sharing screen meanwhile that time, displaying HPFP server.
Okay, sorry. So, okay. So that time, okay, you stopped sharing, is it?
Yes, sir.
Okay.
Fine, sir.
Yeah, I'm sorry, extremely sorry. You should have alerted me that it's that time itself. Anyway, so
actually,
this is the sequence alignment.
With the common definition for missense mutation, so I thought that you are just explaining missense mutation to one and all, and then sir, you just proceeded. So I just bringing you back.
Okay. Now you're able to see the alignment.
Yes, sir. Yes, sir. We do.
And the column where G is absolutely conserved, and now a missense mutation G to K. The question is whether this mutation is disease or neutral mutation. But as far as the evolution of this protein family concerned, at this position, G is absolutely conserved. By virtue of that, you can say that perhaps lysine is going to be harm, is going to do some deleterious impact on the protein, right? But the question of how whether it is really going to cause a deleterious impact or not depends on the strength of the evidence that you have, right? So if suppose you, instead of these many sequences, if you had just 2 sequences, representing this family, that was perhaps is not sufficient enough to say that G is absolutely conserved during the evolution, right? Sequences where G is seem to be occupying the same position doesn't mean that G is absolutely conserved, right? So you need to have a large number of sequences to say so. So this is of sequence alignment, right? How large the data size is, right? That is not it. So you have to also answer a question whether G is part of any catalytic site or a binding site, or is this site structurally important, right? So that these things decide whether this G change to K is really going to impact the structure and function of this protein. So there are various things to be answered. So therefore, so we built a few features. So one is the position specific features. It is all about the how well a given protein, a given amino acid residue is conserved at various positions in the protein. So that is what is referred as position specific features. So there are a few things we could compute. The bioinformatics. There are many topics in bioinformatics which tell you how to compute so-called sequence profile, which goes by the name Gribskor score, right? So, and also there is something, from multiple sequence alignment, you can compute position-specific probability score. So there are ways. So I may not go into the details. So the features are position-specific features. So you can compute how well a given, wild type residue and its substituted residue, which is a missense mutation, is conserved in a given position in a given protein family. Now, the second thing, that structural context. So structural context is whether the position that is there, that has undergone this mutation, is it exposed or buried, right? If suppose glycine is exposed and it is substituted by a lysine, so glycine is now changing to a polar residue rather than a charged residue, positively charged residue, and it is exposed. So the impact will be that much because it's a polar residue, it's a charged residue. Therefore, it can happily stay on the surface of the protein exposed to the solvent, right? So, or it may be protein-protein interaction site, and that basic side chain may form salt bridge with acidic side chain, and therefore it can happily stay there. So there may not be that kind of deleterious impact. But if suppose the glycine is in the buried, well buried within the three-dimensional structure, then changing to lysine can have a detrimental impact, right? A deleterious impact, because now the lysine is a polar side chain, and the protein core is mostly hydrophobic, and therefore you are trying to place a side chain within the hydrophobic core. That is actually energetically very unstable condition. So therefore, it might actually impact the protein fold and therefore the molecular function of the protein, and therefore that might actually lead to the disease phenotype eventually, right? So, and not only that, so the glycine, which is a loop loving residue, right? So when it is changed to lysine, which is actually a secondary structure preferring residue, like such as helix, so there is a clash. So the one, or for example, lysine is changed to glycine, right? So lysine is conformationally restricted, whereas glycine is conformationally flexible. So therefore, there is actually a conformational change in the conformation, extent of conformational flexibility also at a mutation site. So these are the features that we considered and then computed for various diseases and neutral set of missense mutations. So in fact, there is a data set referred to as HumeWar. Okay, so this is available here. So this is the, as I said, if you wish to develop a method, there are certain goal sets that have already been defined. So when you develop your method, so you have to train and then test and then validate using goal set. Then only your paper gets published, before you apply it to any other new protein sets. So our data set has about 13,000 disease mutations and about 9,000 neutral mutations. Okay, so we used again lib-based VM, which is the suite of program available for support vector machines. And we of course used five-fold. We could use five-fold cross validation because there are a number of heads. So we developed eventually, and that is published in Human Mutation Journal. And we could show that these features and the method that we have developed, that named as Hansa. Hansa, it's a bird, right? Hans, it's a bird. There is a folklore which says that if you mix the milk and water and give it to Hans bird, it only drinks the milk portion and leaves out the water. So that means it has a very good discriminating ability. So our Hansa tool also has good discriminating ability between disease mutations and benign mutations. So that's why we came up with that name as Hansa. Anyway, so we could actually show that our our Hansa performs much better than the other methods, which are polypin 2, SAFET, etc., and that is actually shown in terms of this representation is referred to as ROC curve, receiver operator characteristic curve. So what this curve gives you at given false positive rate, that means at given misprediction rate, what is the correct percentage of prediction. So this is, so if the curve is something like this, right? So where area under the curve is exactly equal to 1, that means you are doing the the best prediction. But and if the line is below the diagonal like this, that means your method is completely wrong. So somewhere you have to see above the diagonal. So your curve should look like this. So these are the curves for other methods, but this is the curve for SVM. So you can see that the area under this curve is much higher than the area under the curve for this. So that's how the ROCs are interpreted. Anyway, so we could show that our Hansa is the best model available. That is actually available in this. So I have snapshots for this. So no need to come out of the sharing of the screen. So this is what Hansa looks like. So all you need to do is submit a sequence and then submit the sense mutations, the wild type residue with a gap, the position, and then gap the mutation, whatever the substituted residue. And if you submit, it will give you this kind of an output. This is an example. So whether this mutation is whether it's a neutral, etc., and what are the values of the features, various features that have been computed for that wild type residue vis-a-vis the mutated residue, right? So this is about Hansa. So I would like to acknowledge my two PhD students. Now they are themselves scientists and professors. So Dr. Mohammed Tabez Shamim. So now he is a faculty member in I forgot the name, in California State, in US. He some years he published. He finished his postdoctoral training. Now he is independent faculty. So Dr. Vishal Acharya, soon after his PhD, he joined as a scientist in IHPT Palampur. Now he's, he's a very prolific publisher. He has a number of PhD students under his, belt. So these are the two PhD students. Of course, long time ago, they did work on these two problems. So after this, I didn't get into the machine learning or prediction, development of prediction tools. So my current domain is completely in construction and analysis of protein interaction networks under different contexts. So thank you very much for patiently listening to my this talk at this hour. So if there are still questions, you can ask.
Thank you very much, sir. First of all, I would like to thank you also for sparing your precious time at this moment, especially in evening hours. So there is a question in chat box. I'll read it for you. So is Hansa also suitable for bacterial or other organisms?
One minute. Sorry. Let me open the chat. Okay, sorry. I'm not able to open it. Yeah.
No problem, sir. Is Hansa suitable for bacterial or other organisms proteins also?
Oh, no. See, Hansa was developed for human missense mutations. Okay. But if you, whatever the test and training set that they have used, this is called HumeVar, human variants. Okay. So this is HumeVar data set. So what are the proteins that are considered and the missense mutations that are considered? So they are related to human, phenotypes, so-called benign and so perhaps that's a very nice question. In the sense that suppose you may have a homologue or human homologue in some bacteria or some other organism, a homologue, and it may have the same mutation, but that phenotype we don't know whether that leads to in that organism a deleterious impact or a benign impact. So that is not known. Okay, that is one hand. But if you see carefully the kind of features that we have used, they are mostly on the sequence, that is evolution, that is a position specific preference, and set are structure dependent, right? So you can see the structure, whether it is whether the protein belongs to human and it's homologue in some bacteria or some plant, it's homologue. If it adopts the same three-dimensional structure, that means it is more or less going to have structure. So because we have trained based on the evolution information and the three-dimensional structure, it is likely that I'm saying we have not tested it. It is likely that whatever you have predicted for human system could also be extrapolated to other systems, provided the whatever the three-dimensional structure is highly conserved and the position is evolutionally preferred or not preferred. So these are the two things. So if you carefully see, the features appear to be organism independent, but they are mostly dependent on the molecules, right? So we are not saying that there is a human specific structure impact or a sequence impact is there. There might be, but by and large, it may be, it could also be applied to the other system, but we haven't tried, So that actually it's left. It has to be explored, But for other systems, you don't know. So some mutations are lethal mutations. So they are very easy to predict. But we are talking about the mutations where these mutations don't occur at the catalytic sites or binding sites. They occur where? but you don't know by just mere looking at the position, their position, the type of changes. It is very difficult to say, just by looking at whether they are going to be deleterious, benign. So that is where these kind of prediction tools come into picture. So what is developed for human system, can it be applied to the other system? It has to be explored. So, but we haven't explored that.
Thank you, sir. Thank you, sir, for such giving elaborated explanation. Sir, there is one more question from one of our participants. Can you
Yeah.
please elaborate protein fold recognition based on time reference?
Time reference. Fold. Okay, so what do you mean by time reference?
Gaurav, you can just
In terms
unmute
of
yourself
evolution?
and you can just ask question directly to sir.
So what exactly time reference? Is it with reference to evolution of protein folds, or it is reference to the so-called dynamics of proteins?
Gaurav, am I audible to you? Gaurav, you can just unmute yourself and can ask the question directly to sir.
So let me assume that perhaps bring time reference as we see as dynamics of protein structures. For example, two. So if you, even though we may represent a protein fold as one, static three-dimensional structure, the proteins are actually dynamic entities. So whatever the protein structure that is discovered or that is determined by means of X-ray crystallography, NMR spectroscopy, or cryo-EM, so they are what are referred to as canonical average structures, right? So, if you consider protein, it's an ensemble of atoms. So atoms actually always they are mobile. But even though they are mobile, you can always identify their mean position. So if suppose this is a position, this is the XYZ coordinate, but the atoms move like this, or vibrate, stretch, etc., etc. So there are some motions associated with atoms. So as a consequence of each is atomic motions, so you can actually see that the entire protein structure is also dynamic in nature. So there may be domains moving like this, loops opening and closing, all sorts of motions are involved. So, but all these motions happen more or less like in a harmonic fashion, so to say, about a mean structure. So though we say that's a one structure, stable three-dimensional structure, but there is a limited motion about that mean structure. So that actually could be studied using what are referred to as molecular dynamics simulation. So there is entirely different approach for that. Okay, so that is by solving Newton equations of motion at a given temperature. So there is at a given temperature, there is a certain kinetic energy associated with that. And because of by virtue of the positions, there are some certain potential acting on the atom. A combination of potential energy and energy, which actually gives rise to some motion as far as the atoms are concerned, because each of the atoms experience different forces by virtue of the other atoms within the structure. So that actually gives rise to certain dynamic or dynamical effect of on the protein structures. So that is when you consider a protein fold, we are always assuming that it's a canonical average of all these dynamic substructures. But when you come to IDPs, the story is very different, right? So under physiological conditions, the IDP can assume different conformations, and one conformation to the other conformation, it quickly moves, just so that is what is intrinsically the proteins. That is about the dynamism of the protein structure. Now, when it comes to the evolution, say, for example, if you consider protein family, like aspartic proteinases or proteases, like do, all through the evolution. So when you see, when starting from the prokaryotes to eukaryotes, etc., is evolution happening at the organism level, and as the organism level evolution is happening, so all these molecules are components of these organisms. So when the system, if you consider organism as system, so when that system is undergoing some change, even the components also concomitantly should adopt some changes. So this is what the principle of molecular evolution, right? The organism is evolving, the components of that is the molecules also concomitantly undergo certain changes. And what are these changes? If you look at the level of sequence, amino acid sequence, for example, protein sequence, is with respect to the insertion, deletion, substitution of amino acid residues, right? So if you look at any particular protein family, like a protease family, so you take the sequences from, prokaryotes, you take the sequences from, eukaryotes, and do the alignment, you see certain changes, right? Substitution, insertion, deletions. And if you look at their corresponding protein folds, you may also see changes, So one helix may be 10 residues long in one prokaryotic protein. The same equivalent helix in eukaryotes may be 15 residues long, something like this. Or maybe the loops may be different. Sometimes even the number of secondary structures may slightly differ. So if suppose there is a 4 helical bundle there, it may be like 4 helical bundle with shorter helices, or there may be an extra piece of helix instead of loop. These kind of small, structural changes you might see, and they arise as a consequence of in the amino acid residues. And how these changes are, harbored, that is because now the system is evolving, the organism is evolving, and it has to, and as a consequence, the components, which are the molecules, also should undergo some changes. So this is all interrelated. That if you're talking about, evolution time, so these are the, I don't know what you meant, but I explained in both the contexts.
Thank you,
I
sir,
hope
for
that
such a
answers your
detailed
question.
explanation again, sir. So there is
Thank
one
you.
more question. Checking protein dynamics is microscale or nanoscale, which is more relevant?
That's a good question. Depends on what kind of computer power you have. Okay. So if you say, desktop, even the modern desktop, to for example, a protein of amino acid to do simulations of second. So that may take out a week or month or something. The size of the protein, that is number of atoms in the system. So when you are doing dynamic simulations, it is not just atoms in the protein, but for example, if it is a soluble protein, you have to, create a box of water and put protein in the box of.
I guess there are some network issues because of which sir has left. Sir has joined back again. Just a minute. Hello, sir, am I audible?
Am I audible?
Yes, sir, now you are audible, sir.
Yeah, you are audible. Oh, I was not audible?
Yes, sir.
Explaining.
It was like your bandwidth was very low for some time, so you just got disconnected.
Okay. Now it is all right, right?
Yes, sir. Now
So so what I was trying to say is depends on the kind of computer hardware you have, and whether you have, and the size of the system that you are dealing with for multi-dynamic simulations. So really speaking, it depends on what kind of dynamics, at what level you wish to see. For example, if you consider an enzyme where there is an outer loop and there is a binding pocket, so there is a substrate, substrate enters into the binding pocket and then loop closes over it, so that surrounding water molecules are not entering, and therefore that catalytic activity takes place nicely, right? If you want to do that simulation, like closing, opening, closing, opening, it runs into it's a slow time scale. So it may be microseconds, it can be even longer than that. So it depends on kind of question you are asking. So suppose if your simple your question is simply to identify to study how various secondary structures uh, you know, uh, displace or undergo rigid body motions within a protein fold. So just by at the level of nanosecond should be all right. But if you are looking at domain motion, large loop motions, etc., that might be at longer time scales. You may have to do longer simulations. Suppose some simulations right now we are doing that, so we are trying to simulate a system which is actually a membrane bound system where the substrate percolates from extracellular space through this system periplasm and then goes into the cytoplasm. So if you want to actually, and of course we are applying some force on that, so it is actually a very slow process, so it may get into maybe microsecond level. So for that, we need to have GPU based kind of servers. So it depends on what kind of question you are asking, what study you wish to do, and what is the size of the system that you are, considering. So all these matters. So if you have very huge system and you wish to study the slow motions, like relative motions of domains, large domains like this, or loop motions, or percolation of substrate, etc. So these are at uh, higher time scales, and therefore you may have to choose the hardware accordingly, right? So you may have to use GPU based servers. So these days, so you need not, of course, have a computer server at your place. If you have money, like a consumable money, you can buy this cloud, based services. So Amazon actually is providing cloud services. So you can hire so many GPU nodes with so much of disk space, etc. So I was told recently that you can hire, if you are from an Indian academic institution, Amazon gives you huge discount, and therefore you can you can run your system with minimal cost. So I haven't tried, but I recently came to know. You can try.
Thank you, sir. I'm sure that by today's talk of yours, our participants would have benefited a lot, sir.
Thank you. I hope so.
I'm sure, sir. Sir, I have a small request to you
Yes.
that please share
Yeah.
your presentation with us so that we can also
Sure. Yes.
disseminate the presentation among the
Yeah.
participants.
Yeah. I'll send you by tomorrow. Yeah.
No problem, sir.
Okay.
All right.
Yeah.
With
Okay.
this, sir, with your permission, if you allow, we can close the session.
Thank you.
All right. I thank, on behalf of every one of us, a very thank you to you for giving your precious time with us.
Yeah.
Sir, there is a participant, Nitin Kumar Singh, who has raised his hands.
Okay.
Dr. Nitin, you can just unmute yourself and can ask question if you have any in your mind. Dr. Nitin, am I audible to you? Yeah. Dr. Nitin?
Now it is unmuted.
Yeah, now you are audible.
No, sir. Any query नहीं, कोई query नहीं है, sir.
All right,
Sorry,
all right,
sir.
it's okay. No problem. By mistake, sir, he raised his hands.
No, it's
He
okay.
doesn't have any query. All right, sir. Thank you, sir. Good night, sir.
Yeah,
Thank you,
thank
sir.
you.
Sure, sir.